now.
Well, welcome everyone to this, the latest in our series of webinars. We host this event as the others under the banner of the Centre for Intelligent Design in the United Kingdom, and we're really pleased that you've joined us for this session. Over the past many months now, we've been keen to present a whole sequence of different topics to provide an understanding of aspects of design in nature, and asking questions about design and considering the related evidence is not only in the healthy pursuit of scientific truth, it's the way that we have begun to really emerge maturely in terms of scientific understanding over the past few hundred years. Now today, this is our 15th such webinar, at least in this format. You can find all the previous recordings on the C4ID YouTube channel. We've focused on a whole series of things ranging from design and virology at the beginning of the pandemic through a whole range of other issues, including one previous episode where we looked at some aspects of the material that we're going to be considering today, and intriguing it will certainly be, as you'll find out. We've looked also at various forms of bias within scientific thinking. We've hosted people such as John Lennox and Mike Behe, Steve Meyer, Jim Tour, Doug Axe, Mike Denton, and Gunter Bechley. Today's lecture, like Gunter, is also from Germany, and that is Professor Peter Imming. And the title of his talk, I think, is intriguing: The Origin of the First Cells. And the subtitle, Soups, Pizzas and Minds. So I guess you'll be as intrigued as I am to find what he's going to say about that. But certainly how life on Earth first began certainly continues to be one of the most interesting and difficult conundrums to really address. There's a burgeoning origin of life scientific literature. Many extravagant claims are often the outcome. Extrapolation is inclined to be wilder here than in many other areas of scientific endeavour. And in this presentation, Peter's going to be considering various approaches to the ultimately unobservable beginning of life. And I think he's also going to dip into some non-reductionist, non-theistic philosophies that have been introduced into the thinking by way of explanation. So this should be a very interesting, perhaps challenging session today. Peter Imming earned degrees in pharmacy and chemistry, as a PhD from the University of Marburg in Germany, and after a research stay in Oxford, he held positions at 3 German universities. And since 2004, he's been Professor of Pharmaceutical Chemistry at the Martin Luther University in Halle-Wittenberg. His research presently focuses on designing, synthesizing and testing antimicrobacterial compounds and on anti-infective medicinal plants from Africa. And along the way, he's very interested in this topic that we're going to be considering today, because it has an impact on worldview, his own view being based on the premise of a creator. Now, as usual, after Peter gives his presentation, I'm going to ask my colleague David Williams to moderate the question session. So please make use of the chat facility on YouTube to post your questions and comments as we go along. And in the meantime, Peter, it's over to you. It's a real delight to welcome you to this session and to the UK, albeit virtually. And perhaps I can invite you now to go ahead and tell us about this topic, the origin of the first cells. Thank you very much.
Thank you, David, for the invitation and for the introduction. Welcome, everybody, from the city of Halle in Germany, about 1 hour south of Berlin. If you don't know where Halle is situated. Of course, I would have loved to have returned to the UK for this occasion, and of course for a couple of more days in what I have come to like a lot since more than 30 years ago. I did a research stint at the University of Oxford. Well, let me just share my screen so that you can see the slides I've prepared. Okay, David, if you could just give me information if everything is okay technically. Slides.
Yes. Yep,
okay.
Good.
Okay,
good.
Very good. Just to make sure. Right. The origin of the first cell or first cells, pizza, soups, and minds, is the question about the transition of dead matter to living matter. This, of course, already implies some consideration which we've made, that we think that dead matter perhaps came first and living matter second. Anyway, this is what the whole question is about in scientific terms. And the scientific disciplines involved are chemistry, which is my topic, although I'm not an origin of life researcher, and information theory. Chemistry and information theory are the leading sciences in the question on origins of life, origins of first cell. I'd like to quote the famous origin of life researcher, chemist Stephen Mann from the University of Reading. Almost 10 years ago, he said that a study of biology offers no illumination on the origin of life. Well, that's a rather dogmatic way of putting it. But anyway, we are basically talking about life before the first cell, so life before actual biology set in. In other words, we're asking for the origin of biomolecules and biomolecular networks. To use a metaphor, we're asking for the origin of the hardware we consist of, and so many other beautiful living beings on this planet. And we also ask for the metaphor software, the biochemical and cellular organization. How did this all come about? We can study it in its present status, so we know a lot of detail about biochemistry and physiology, but we'd like to know, okay, how did this all come about? How was this transition from, if you look at the picture at the back, this transition from purely physical structures to biological structures, like as in this jellyfish, how this is basically come about? This, of course, is just a symbolic picture. We're asking for, like many people have put it, the hypothetical pathway from dead matter to first cells. This picture shows you like a simple way of putting the question in a little bit more detail. We assume that there was a primordial atmosphere consisting of energy and, let's say, simple molecules, which then somehow turned into a primordial soup or pizza. I'll explain the terms, which are not invented by me later. This primordial soup is supposed to have organized itself later to proteins, nucleic acids, all the building blocks, or some of the building blocks that we actually need for a cell. So we're asking for, how did a protocell, protobiont with a cell membrane, how did this first come about? And then, of course, the study of biology, of evolution, and so on would take over. But this is not what we're going to look at. We're just going to ask, how did a first cell or first cells come about? The next slide differentiates the question again a little bit. We are talking about origins, but we're also asking what was first at the origin. What was the first origin of origins in this context? Most hypotheses and theories that we know of fit into one, two, or three classes. If I return to this picture, this is from a paper that appeared in 2020, a nice illustration. I find there are people that talk about replication being first, symbolic molecule, RNA. Some people would say, no, metabolism was first, energy consumption, energy generation, metabolic transformation of molecules into other molecules, the reverse tricyclic carboxylic acid, tricarboxylic acid cycle is what we're going to look at. And I will not tonight, partly for reasons of time, look at the compartment idea, although I will touch upon it briefly during the talk. So what was first? Was there replication? Was there metabolism or compartments? In the end, we'll need all three, let's say, parts or constituting elements of cells. The question is, what occurred first? Well, most people would, like I said before, fit their theory, their hypotheses or scenario into one of two classes. And that is what I'm going to use to structure this talk. I'm going to use this as a structure of this talk. One of two classes of hypotheses on the origin of life, or to put it less philosophical, because life, of course, is a very complicated concept, origin of cells, first cells. One class of hypotheses and scenarios would say, well, it was polymeric molecules that were formed first. Polymeric molecules which were not just polymeric, but that had the ability to replicate, replicator first, genetics first, RNA world, primordial soups. This is all different expressions for this type of hypothesis. And we'll look at that in some detail. Next to that, or parallel to that, there are hypotheses that people call metabolism first scenarios. Metabolism first scenarios. Some people call that primordial pizzas, because most of these hypotheses need a surface where molecules can assemble and react with each other. But the basic chemical idea behind this first origin of origins is, first there were energy-driven networks of chemical reactions with lower molecular mass molecules, not polymers, but lower molecular mass molecules. You will see references at the bottom of many of my slides. There are 2 slides at the back with all the references in detail for you to be able to look that up, and I'm sure there will be a way to let you know the references in detail after you've listened to this talk. We'll start at replicator first hypotheses, or scenarios, as many people put that. This is an illustration, just an illustration, and I have to perhaps apologize at this point to chemists, or people that know a lot about chemistry in the audience. I will also apologize to people that know little about chemistry, because for a talk like this, well, I have to take account of the fact that many people chose not to continue chemistry at school. And very often when I identify as a chemist, people tell me that they did not follow up with the subject even as early as 8th form or something. I'm of course very happy as a chemist to hear that. Well, I'm not actually, but I have to take account of the fact that chemistry is not on everybody's mind. I repeatedly meet people that do not know the periodic system of the elements by heart, but they seem to somehow survive. Quite a mystery for me as a chemist. Returning to our subject in hand, replicator first, just an illustration. For a chemist, perhaps too simple, but for all of us, perhaps a good way of getting into this of an evening like today. First, there was a primordial mixture of building blocks, smaller chemicals, smaller molecules, and we just assumed they were there. We could, of course, ask where did they come from, or we have to start somewhere. So we'll assume there were building blocks, which I've chosen to symbolize by letters. By chance, they assembled to a big molecule, a polymer, a molecule consisting of smaller molecules, each bound to each other, like a chain. And we'll call that, by way of analogy, start life. Now, this, of course, had to replicate this molecule. For example, RNA or a protein, in chemical terms, had to have the ability to replicate. So there are now 2 start life molecules. Somehow, this polymer acquired or had the ability to replicate. Then it had to mutate, because we have to go on to like the diversity of life, and polymers, RNA, DNA, carbohydrates, and so on, which we see in today's cells. It had to be able to mutate, which it did by chance after a while, from start life to smart life. And then smart life was more stable than start life. It was selected, and of course it again had to replicate. Mind you, biologists in the audience, when I say mutation and selection, I mean it, but I don't mean the kind of mutation and selection which you find in living organisms when you look at evolution. This is not the case here because we don't have the organisms that can like do the mutation or selection. We are talking about, let me put it like purely physical chemical processes. From here, the replicator first theories assume that somehow compartments formed which included the polymers, like the smart life, another smart life, and also metabolic circles, as we will be talking about in a few minutes. And this is assumed to be a kind of protocell, something that was able to somehow change into a cell like we know it today. Of course, I also needed something like a lipid vesicle, a lipid layer, or some type of compartmentation. This illustration is summarized here. It's the replicator first type of theories, and I've modified this illustration after a paper Robert Shapiro, the late Professor Shapiro, published many years ago in Scientific American. The illustration as such is still holds. So now let's think a little bit about the replicator first theories. Are they plausible? Are they plausible? Actually, many people think they are not. Of course, there are also researchers that think they are plausible. Well, let's have a look at why researchers, for purely scientific reasons actually, think that replicator first theories are not plausible. You've already noticed that very often I had to say, and this is not my fault, I believe, that somehow this happened or somehow this turned into something somehow. So it's not quite sure how and why this should have happened. Shapiro himself put it like this: the spontaneous appearance of any such replicator without the assistance of a chemist faces implausibilities that dwarf those involved in the preparation of a mere nucleotide soup. Remember, we didn't even discuss how the monomers, the nucleotides, if it was RNA or DNA that had formed by themselves. DNA, RNA, proteins, and other elaborate large molecules must be set aside as participants of the origin of life. And he's joined in this skepticism by a recent review paper by Bukowska and others, 2020. Just a quote from this paper. I cannot go into depth now, but I hope this will be sufficient. Sequential chance event scenarios cannot achieve kinetic coupling between subsystems and thus lack one of life's critical features. They're also unlikely to occur without human intervention, the chemist, which Shapiro said was necessary to do this. So the kinetic coupling between subsystems that, like, to put it simply, everything moves forward at a pace or reacts with each other and among each other at a pace which is in sync with the whole system. That's just not, there is no way how this should come about by itself. The famous Professor Eschenmoser in another review paper actually wrote that neither the formation nor the autocatalytic replication of an informational oligomer under potentially prebiotic conditions have been convincingly demonstrated experimentally. So he says experiments don't show that it works. Now, that, of course, for many people, that is a bit confusing because many people have heard of the Miller-Urey experiments, which started in 1953 with Stanley Miller, shown here, showing that the production of amino acids is possible, and even of peptide chains under possible primitive Earth conditions. So people would say, but there is the Miller experiment. Didn't he prove? Okay, let's have a look at what he actually proved and how Miller himself, many, many decades later, thought about his own experiments. What he did was, you see the experimental apparatus of his original experiment behind him. What he did was he exposed a mixture of simple molecules, small molecules, ammonia, carbon monoxide, methane, and so on, to electrical discharges, so to a lot of energy. And then he collected what reacted in this reaction vessel with water cooling and collected whatever formed down here, shown in violet. That's what he collected and then removed and analyzed. Water was also included in this whole process. And this experiment actually was repeated probably hundreds, maybe thousands of times with under different conditions, with different mixtures of chemicals. But the result was always maximum. The maximum result was always what I'm going to explain to you on the next slides. So this was about the production of amino acids under possible primitive Earth conditions. What he collected at the bottom of the experimental setup was an oily brown mixture of many chemical compounds. And indeed, as he had assumed, he was right in that, amino acids formed. Actually, more amino acids than we need for the proteins that presently constitute living organisms. Some amino acids did not form even later. Some amino acids were elusive under this type of experiment, but perhaps if the composition would be changed, then they would also be formed. The main flaws of this whole setup were the following. One, the composition of the mixtures was not comparable at all with the molecular composition of living creatures. The brown oil which he collected it got nothing to do as a whole with what a living organism can be reduced to. And more importantly, the more important flaw of this experiment was that, of course, the next step after the formation of amino acids would have to be the polymerization to give peptide chains, because that would be the logical and necessary next step towards biomolecules, in this case, proteins. However, the Miller mixtures, not only Miller's mixtures, but all mixtures that have since been analyzed, contain monofunctional compounds preventing chain formation. So the Miller experiment and Miller mixtures are actually a blind alley, including recent reinvestigations. There have been lots of reinvestigations, and they're all blind alleys. They don't lead further. They don't lead to biomolecules, to what we need to constitute living organisms. Why is that? Excuse me for using another colorful illustration to explain that. Peptide formation through polycondensation of amino acids needs, of course, when you want to do a polymer, you need to have two connecting points for each monomer. Each monomer, which is supposed to polymerize, like shown here, needs 2 points where it can connect to another molecule. It could also be like this, just one type of molecule, for example, an amino carboxylic acid, amino acid, like we have forming proteins. They have an amino group and a carboxylic acid group, which can form basically endless polymers. But the problem is that in Miller mixtures, always monofunctional molecules form, lots of monofunctional molecules. And if you have a mixture of monofunctional and bifunctional molecules, you'll only get very short chains, because the moment a monofunctional molecule reacts with any of the bifunctional ones, that's the end of the chain. So it's a blind alley. Stanley Miller himself, when he was interviewed a few years before his death, he died in 2007, Miller himself explained or expressed himself like this. And I'm quoting for a book by Horgan. Miller remembered predicting shortly after he had done his first experiments that within 25 years, scientists would know for sure how life originated. Well, the 25 years are over, he dryly commented. None of the current origin of life hypotheses seemed to convince him. He called them nonsense and chemical mind games. So that is what Stanley Miller thought about the experiment, which he had quite cleverly set up, but it proved to be, scientifically proved to be, a blind alley, a dead end, when trying to explain how, in this case, proteins were formed. So like I said, many people, obviously for scientific reasons, have become disappointed by the replicator first theories, which we have touched upon in the past 5 or 10 minutes. We'll now have a look at the metabolism first scenarios. Energy-driven networks of chemical reactions were formed first with lower molecular molecules, primordial pizzas, because they need surfaces for these scenarios. Again, we start by a primordial mixture of building blocks. We always need that, of course, wherever it comes from. And we keep it very open as to what this mixture could have been composed of. Of course, we try to inform what we put in that mixture, either theoretically or experimentally. We try to inform that by what we know of the primordial Earth, by geological hypotheses or investigations or whatever. And this scenario says that some of the molecules got trapped in compartments. They got trapped in compartments, and some of the compartments contain molecules which could react with each other in a kind of cyclic fashion. So simple for chemists, but for the non-chemists among us, this doesn't mean that they form chains. This is not like a polymerization, not a chain formation, but the arrows in this case mean that B can be converted to C, C can be converted to A to D, D to E, and the E can then be reconverted through a chemical reaction to B. And the whole thing generates and consumes energy. So it's like a small reactor, just symbolically. Some of the compartments were not able to build up such metabolic circles, so they were selected out, just chemically, not biological. And after a while, the initial compartments containing reaction cycles got more complicated, more complex cycles developed. And this then, of course, had to match with the replicators, which in these scenarios come second or were developing in parallel. Anyway, ultimately, the metabolic cycles, the reaction cycles, and the replicators, of course, have to have combined, because we want to understand cells like we can observe in our present world, which contain both. This next slide again shows the replicator first scenario at the top and the metabolism first scenario. Very simply put, I apologize again, but I hope this will be helpful to keep track. Metabolism first scenario. So let's have a look at a metabolism first example. How does this look? What does this look like in chemical terms? Now comes more chemistry, still not enough chemistry for the chemists among us, but still. An example of which is very often discussed among metabolism first scientists, people that follow in their research this scenario. An example which is fairly often used is the reverse citric acid cycle, reverse tricarboxylic acid cycle. So that's the same, RTCA and reverse citric acid cycle, that's just two different words for the same chemical reaction cycle. This reverse citric acid cycle with non-enzymatic catalysis, because we assume that replicators like enzymes will only be formed later, is with non-enzymatic catalysis is discussed as part of the chemistry that may have led to the origin of life of cells. Why the reverse citric acid cycle? Because, of course, inorganic carbon fixation is essential to sustain life on Earth. We need carbon for living organisms. Organic molecules consisting of not only, but most importantly, carbon atoms are essential for biomolecules. And the reductive tricarboxylic acid cycle is supposed to be, or is, one of the most ancient carbon fixation metabolisms. This is what it looks like. This is what the reverse citric acid cycle, RTCA, looks like. You see it's a number of molecules, citrate, oxaloacetate, malate, fumarate, and so on, being converted into the next molecule in the cycle, and this could go round and round, actually in both directions, depending on the concentrations of the individual molecules, and also depending on the type of catalysts, enzymes, or whatever around. Of course, this is also connected with more metabolism, like shown here, but I won't go into all detail. Why do people think that this is a plausible reaction cycle to explain the origin of life? Because this cycle, nobody believes that this cycle formed just spontaneously, however long ago. Nobody believes that this whole cycle formed spontaneously. So the question is, can we devise or experimentally set up a simpler cycle, which could then have changed into this cycle? I avoid the word evolve because that smacks a lot of biology. We are not talking biology here, just chemistry. Of course, biology is also a lot of chemistry, but I hope you get me. So the question is, could this have evolved from simpler beginnings? And there was actually a paper in 2018 published very prominently in the journal Science of the discovery of a reverse TCA cycle with a reverse reaction of a citrate synthase in a certain bacterium, which occurs in hot environments, thermosulfidebacter species. So people thought that, like, okay, this is perhaps an indication that with a simple organism like this, we have a reverse tricyclic acid, tricarboxylic acid cycle, excuse me. Perhaps this is another indication that this could have been one of the cycles that constituted a first metabolism, which then helped produce the first protocells. However, even with this finding, you know, it needed an enzyme for this. Even with this finding, let's have another look at this cycle. You know, even if we think of a simpler circle, still it would need a lot of concurrence. What do I mean by that? A lot of molecules with the right concentrations have come together, and it not only needs the molecules whose formula you can see here, it also needs the numbers. The numbers, black dots with numbers, they are catalysts. Enzymes are something that is not an enzyme, but a catalyst. And you need all this, or at least some of this, at the same time concurrently. Otherwise, the whole thing will fall apart or will not even form in the first place. We actually need very specific catalysts, because quite a few of the reactions, if you look at some detail now, so quite a few of the reactions in different corners of the cycle are chemically very much related to each other. For example, this dehydration, or if you look at it the other way around, hydroxylation, dehydration. This is fairly similar chemistry on fairly similar molecules, but not the same molecules. So the catalysts have to be very specific, otherwise the whole thing won't work. If you have a catalyst that speeds up all of this, the whole cycle breaks together. You don't get it running. And there are, of course, parasitic side reactions. There could be reactions can occur, certainly in a prebiotic environment, that are parasitic on what's happening here. then there is these enzymes. We need catalysts, and in the end, we need enzymes. So where's the genetic basis for the enzymes? Because it must have formed relatively quickly after the metabolic cycle came about. We need that all at the same time. So the evolution of such a cycle from simpler beginnings is not plausible. As Mukovska in their review write, the need for metabolic pathways to work together as a whole means that constraints on the non-enzymatic chemistry of one reaction or pathway become relevant to the others, and ideally all must be considered simultaneously. I've made a list of why metabolism first scenarios are supposed to show pre-Darwinian evolution, like many people put that as evolution, but that's just I think just a debate on terms. But all these scenarios show major issues. I've put them together in a list. No practical chemical substrate for such a cycle is known. Well, what about the tricarboxylic acid cycle? Yeah, but that is not the primordial cycle. That is something which we know exists and it works in live cells. It doesn't work outside live cells. Experimental data supporting the main hypothesis on which the model is based are lacking or preliminary. Just a lot of would-be, perhaps, might, could have, must have, but there is no must. There's just chemical laws, isn't there? The source of energy is questionable or not specified. The energy is either too low or too much for what's discussed here. The chiral question is not addressed or not considered as an issue. Just walking back to the depiction of the cycle, you have centers of asymmetry, like here at this point. For example, again here you have stereogenic double bonds. So all these stereo factors are not considered or addressed, like I won't in this talk tonight. Multiplication abilities are not truly present or unknown. Heredity is not clearly demonstrated or absent. How does this cycle replicate? A plausible site on early Earth is unknown or not specified. The system is unstable, and the evolutionary path to it and from it seems either complex or questionable. Now, this is actually not my word. This is not informed by my being a Christian. This is informed by Tessera being informed as a chemist and informed by my being a chemist, and so many chemists would agree on that. Staying with metabolism first, let us have a quick closer look at hydrothermal vents and membrane bioenergetics. That's another scenario which has been discussed quite a lot during the past 10 years, perhaps even earlier than that. This is a scenario which belongs to the metabolism first theory group. And as an example, I quote a relatively old paper, Cell 2013. However, this was published in Cell, a very prestigious journal again. And this paper was quoted more than 200 times by people active in the field. So it's quite it's a pivotal paper on this whole topic. The focus is on energetics and metabolism first theories, rather than structure and information. And let's have a quick closer look at chemical detail. So more chemistry for a couple of minutes. This is figure 1 from the Cell 2013 paper, which I selected for the purpose of this talk. So what you see here is a kind of membrane. Of course, it's not a membrane. It's an iron nickel sulfide cluster, which is like a surface or a barrier between outside, where we have protons, ferrous ions, and carbon dioxide, and inside, this is not an actual cell, this is an idea of how something that preceded a cell could have looked like, and inside we have metabolic processes creating energy. The metabolic process consists of the fixation of carbon dioxide to give a thiol ester, which is then converted to a mixed anhydride of acetic acid and phosphoric acid. And this is then turned by X, whoever X may be, that doesn't matter for our present discussion, to acetate. So this is one of the depictions shown in this paper, possible stages in early bioenergetic evolution. And this is supposed to show a chemical scenario preceding the formation of a protocell. So when I look at this as a chemist, I should mention that FD minus is ferredoxine. Actually, ferredoxines are proteins, so this doesn't talk about the protein part. Ferredoxines are ferrous sulfide cluster proteins. You need protein for that, but this is not considered here. It's just assumed that it's there. Anyway, if you look at this with a chemist's eye, you wonder, because it says that pH is 9 or higher. At pH 9 or higher, this type of molecule, thioesters, are not stable. And this is even more true for the mixed anhydride. They're not stable at pH 9. Okay, maybe there was no water. Another assumption, there was a surface. You see, you have to assume a lot of things to make this plausible. A lot of things which are definitely not do not come into existence by a simple process. Some of them seem to be nigh impossible. And this is another figure from the paper where they say that, okay, then there was catalysis, genes and proteins are sometimes suddenly in the picture. So ATP, suddenly there is ATP. Where do the genes and proteins come from? And what do the membrane and constituents come from? Because you see these black lines, they are supposed to be membrane constituents that somehow is concentrated at the surface. Where does the ATP catalyst come from? All of this is just assumed. If you look at this paper, actually pH 9 is also not compatible with ATP stability. If you look at this paper, it's highly suggestive, has very creative illustrations, and it is full of hypothetical formulations, lots of could, may, might, would have. So the widespread skepticism towards metabolism first scenarios applies to the hydrothermal vent theories too, which is not just my private conviction as a chemist. Other people would fall in with that. There are other scenarios and open questions. There are RNA wall scenarios, amyloid scenarios. They actually belong to the replicator first theory. So all the considerations and flaws, which I've tried to demonstrate quickly for the purpose of this talk, when we talked about replicator first theories, apply to RNA wall scenarios the same way. There is panspermia, where people say that life came to Earth through extraterrestrial organic molecules, but that just shifts the physical-chemical problem to somewhere else. It doesn't change. I mean, we don't believe the chemistry and physics is different in the universe in different places. There are different physical and chemical influences and constituents, but the chemical and physical laws are the same wherever you go. That's certainly what everybody believes. There is scenarios which work with lipid vesicles and micelles, which mainly for reason of time I've skipped for tonight. Computational simulations, for example, hypercycles, dissipative structures, which Ilya Prigogine brought up, many from thermodynamic considerations. There are also lots of questions, open questions, which I won't touch upon tonight. For example, time. Biomolecules decompose quickly. So when people say that, oh, there was a lot of time for replicators and metabolisms to form, I would say, no, there wasn't, because molecules can't wait for each other for 1,000 years or 1,000,000 years, they decompose quickly, or especially if you have all the fragile structures, cells, for example, and reaction cycles and biomolecules, they're not stable. They can't wait like stones can perhaps wait for millions of years. I didn't touch upon the origin of homochirality and upon the origin of information. Information as in genetic and other biochemical codes. There's the genetic code, the epigenetic code. There's the code of the immune system, kind of chemical carbohydrate or protein carbohydrate code. So all of this has led many people from whatever background, from whatever worldview, to think about alternatives. The idea, common idea of the replicator first and metabolism first scenarios was that we try to build a cell from basic chemical building blocks. We start small and we try to build big. This, of course, makes sense, scientifically speaking. However, the alternative approach has become more and more popular in past years. What's the alternative approach? It's that we look at the cells which we can find in our day and reduce the simplest cell that we have, reduce that experimentally to find its essential parts, then rebuild it on this basis, and thus perhaps get nearer to an understanding of how the simplest cells could have formed. So this is this top-down approach involves using metabolic and genetic engineering techniques to impart new functions to living cells, whereas the bottom-up approach involves creating new biological systems in vitro by bringing together non-living biomolecular components, often with the aim of constructing an artificial cell. An example of the top-down approach is a major research project. There are quite a few examples. I'm quoting the German-based research project MaxSynBio, which has the objective to understand the underlying principles of life by building cellular structures from scratch using inanimate biochemical building blocks. It is our hope, they say on their website, that the insights will help us to understand the miracles of life. So what they want to do is build a cell. For that, of course, you would like to know what's a minimal cell. The MaxSynBio and other consortia optimize building blocks of a simple cell. And then they want to put this together to a minimal synthetic, fully synthetic system. The goal is to be able to do that in 2028, the latest. You can read that in publications by Pelletier and Schwille and others. Actually, Craig Venter famously also does or did that. He created a synthetic cell, which he called JCVI Syn 3.0, which was made by copying an existing bacterial genome and transplanting it into another cell. And this synthetic cell contained 473 genes. According to his studies, 375 genes, as in protein coding sequences, seemed essential. Well, that's still a lot. So yeah, let's have a look at what we're facing here and how far we've got. Origin of life faces a lot of problems. Well, that's nice, because that's scientists are looking for problems which we can try to solve. So that is not, it's not a problem that there are problems. It's just that we need to define the problems, of course, so we know what we want to study or understand. The problems are all symbolized by question marks. If I just quickly explain this slide as a whole, we want to understand how we can go from primordial soup or primordial something to a minimal first cell, either bottom up or top down. Understand this, going from left to right would be the bottom-up approach. We need amino acids, we need nucleobases, we need ribose, lipids, vitamins, and so on. We need a lot of structure, including polymers like proteins, RNA, DNA, but we don't need any odd protein. We need functional proteins that can do the work in a cell, in a minimal first cell. For that, we also need a genetic code, and the genetic code, of course, needs to match the functional protein formation. It's like going from structure to information. Or if you look at the bottom, as for constituents, we go from monomers to polymers to functional polymers. In a metaphor, we could call, excuse me, we could call them machines in order to build a minimal first cell. And I hope I'm not going to surprise you, but I think that it is absolutely possible that during your and my lifetime, scientists will be able to build a minimal first cell from like scratch, from building blocks. I consider that totally possible. But mind you, if scientists were able to do that, to perform that feat, that would show that they are very creative and they would have had a very good plan and very good experimental conditions and abilities. It wouldn't prove that a minimal cell can form by itself. It would show that people with enough time and cleverness and the right chemicals and a particular goal which they want to approach can do this. So if people succeed in building a minimal first cell, that would, in my terms, actually be an example of creation. However, I'm now going to change a lot of the question marks to exclamation marks, as you can see here. We don't only have problems when we look at the origin of life and how can biomolecules form. We know a lot. We know how we can build functional proteins. People do that all the time, either in the lab from amino acids or using organisms, and we can also do that with RNA and DNA. You can build many proteins, but you have to, of course, do that not by mixing a couple of chemicals and hoping that they will find each other. Somehow you have to have the right building blocks and put them together in the correct sequence, time sequence, and you have to purify in between. So yeah, we know how to do functional to make functional proteins, how to make DNA and RNA. We know how to make membranes and fatty acids and even carbohydrates, even though that is the most difficult chemistry of it all. So all the exclamation marks are not problems, they're knowledge. We know how to make all this. So science leads us to circumstantial evidence for the necessity of creativity for the assembly of living matter. It does need a chemist. It does need planning. It does need that you know what you want to do. And that is why in the past years, quite a number of prominent people in the field have asked for a radical rethink. A radical rethink is necessary. It's actually a quote from a kind of review, Cronin and Walker, which appeared in 2016. The quote I've included in this talk is, "Progress will be made by challenging all historical prerequisites assumed to be important in the origin of life. For this to be possible, researchers must challenge the current models and historical approach and be willing to work together across disciplinary boundaries to see if a process-based model can be used to understand and control the transition from inorganic matter to biology." So control the transition, process-based model. We have to challenge what we've been doing since like 50 or 60 years, looking for the origin of life. We need to try to create life in order to understand the origin of life. As Cleves and others put it in another review, the origin of life remains unexplained despite decades or perhaps centuries of research. It is impossible to predict how soon the problems prebiotic chemistry addresses will be solved, it seems likely that in the near future, chemists will assemble living organisms de novo, creation of life, in order to understand the origin of life. Stephen Mann put it like this in Angewandte Chemie 2013, addressing questions concerning the transition of inanimate matter to the living state necessitates a new paradigm in chemistry that moves away from a radical form of reductionism to a more integrative, reconstructive approach. An overarching objective is the design and construction of non-equilibrium chemical microsystems. So all this reductionism hasn't worked. We need to become more holistic. Sorry, this is of course a very a word that has a lot of connotations, but it's the word that comes to mind quickly. Eschenmoser also said this, putting it like this: the quest is to think of, synthesize, and study adaptive and self-sustaining chemical systems, and in the perhaps very long run, to create what will amount to elementary forms of artificial chemical life. Research toward the creation of artificial chemical life will be an important, if not the most important, empirical source of knowledge for our eventual comprehension of life's origin. So these people, obviously, even though they put it in different terms, put mind before matter. You need mind for mechanism. The origin of life was, it seems, from the scientific evidence we have gathered during the past decade, the origin of life seems to be not something just causal, only causal and mechanistic. Though, of course, it was accompanied by a mechanism, a chemical mechanism, no doubt about that. But that's just not sufficient. It needed organization. Note how often the term self-organization comes up in discussions of origin of life, if you follow the literature at all. Origin of life needed integrative management, construction, attention to detail, creativity, like many people have put. So acknowledging this evidence, it's not speculation, it's evidence that this is needed, that something like creativity is needed, opens up two choices. Creativity is obvious and observable. It's not something we like purely hypothesize. It's observable, both by intuitive and by scientific examination of the cosmos and its details and of cells, for example. It is not a gap of our understanding. It's an empirical and logical observation. The more we know about living organisms, the bigger this evidence for creativity. The more we study the whole subject, the more this comes to the foreground. So the question is, who do we think capable of having original creativity, impersonal, purposeless matter, or a personal, purposeful God? Well, it's up to everybody's personal choice to answer that question. And perhaps there is a mix of two which we could discuss, but anyway, this is basically what we have to consider. Let me close by saying that this whole discussion, which I've tried to follow for decades, for me is a corroboration of a Christian premise. It's not a proof of God or anything. I don't imagine God as like a master chemist or something, although he certainly knows about chemistry. Still, it's a corroboration of a Christian premise. The Christian premise is formulated in the Bible, for example, in a letter to the Romans, as follows: that which is known about God is evident within them, within humans, for God made it evident to us, to them. For since the creation of the world, his invisible attributes, his eternal power and divine nature have been clearly seen, being understood through what has been made, including cells. And when we think about how could these cells have come about, or in the Gospel of John, right at the beginning, mind before matter is famously formulated as follows: in the beginning was the Word, and the Word was with God, and the Word was God. All things came into being through him, and the Word became flesh and dwelt among us. Well, this corroboration of a Christian premise please do not misunderstand me. This does not abolish interest and the scientific inquiry into the biochemical status and origins of cells. It is just that it seems mindful input must not be excluded when investigating the origin of cell. We must not be stubborn, if you excuse me putting it like this, we must not be stubborn and excluded for reasons which are certainly not informed by science. So that is actually the end of my talk. I will put up the references so that they will be visible in the recording, or you can take a screenshot or something if you want to know which references I've used. It's 2 pages of references. This is 1 page, and this is the 2nd page of references. And I thank you very much for your attention.
Good, thank you. Over to you, David Williams. Thanks, David.
Peter, thank you very much for that. That was a fascinating talk. So thank you for that. Let me just see if I can sort out the spotlight system here. Okay. Just a note to everyone, if people would like to ask any questions, and now's the time to type them in the YouTube channel. I know there's some in there, and I'll work through those now with Peter, and then if there are developments or more questions, we can take it from there. The first question for you, Peter, is this. What do you think of the idea of mitochondria, or a mitochondria, being swallowed by a prokaryote and becoming a eukaryote? And if so, has this been observed? I think the question uses the word swallowed. A better word might be engulfed, although it probably may mean the same thing. But so mitochondria being engulfed by a prokaryote and becoming a eukaryote.
Well, that of course is beyond the scope of the talk which I was giving tonight, beyond the scope of the question of the origin of first cells. It's like a little bit further down or on the road to living organisms. There are a number of biochemical details which make me and other people doubt that this actually happened, because mitochondria indeed look like they could have been small cells of their own, but there is a lot of detail which I can't enumerate just from memory. A lot of chemical and biochemical detail which makes it very doubtful that in actual fact mitochondria existed on their own and sort of combined with simple cells. But that is actually not the question, which, if you forgive me not giving a full answer, this is actually not the question which we have before us. This is an interesting question for sure. Even for a pharmacist like myself, I'm basically obviously a pharmaceutical medicinal chemist, because it also means, for example, what do I want to do when I want to use particular drugs, for example, drugs that interfere with energy metabolism. But basically, if I look at the drug discovery literature, I can't remember of a single instance where people have used this mitochondrial engulfing hypothesis as helpful for medicine or the development of drugs. So an incomplete answer.
No, that's
Sorry
fine.
about that.
That's okay. Thank you. Next question is this. Why does it appear that mainstream science is so confident that self-assembling peptides are the answer to the origin of life? And what do you think, and if so, what are they? What are the shortcomings in that theory?
Oh, it's a really difficult question to answer. Why does a theory, even though its origin originator, in this case Stanley Miller, doubts it a lot? Why does it persist in textbooks and even in research articles? Is it laziness, or is it that people have arguments Stanley Miller himself has overlooked? I am not absolutely sure about that. I'm sometimes inclined to think that people just don't look at the chemical detail. I hope I'm not stepping on too many people's toes, but sometimes I'm under the impression that biologists, much as I admire their science, to be honest, don't like to care about chemical detail. And in this case, like I showed, there are these monofunction molecules, which Miller himself, of course, knew. And they make this blind alley. So you need to look at chemical detail when you look at could proteins have formed by this type of chemistry. And perhaps there is a barrier between biology and chemistry that we don't like to look at detail in each other's worlds. I don't want to go so far as to say that people don't want to give up a theory if the alternative is something non-reductionist, perhaps even theistic. There may be people out there that shy away from theistic hypotheses, but I don't want to go as far as to say that people do that deliberately because they just don't like something. Actually, famous philosopher Thomas Nagel does this. He's very honest in one of his books, saying that looking at the status of origins, he feels that a theistic explanation could be a good one, but he doesn't like it. He says so expressly in his book, Mind and Cosmos. That's very honest, and I admire him for that, but that is, of course, not a scientific argument.
Yeah. Okay, next question from one of the viewers is this. It's talking about the Miller-Urey experiment. It says the starting point involves, quote, complex, close quote, chemical compounds. How did they form and in what quantity?
Yeah, that, of course, is a question with a lot of answers, because one would have to discuss that for ammonia, for carbon monoxide, for oxygen, for all components that Miller and others have put into those Miller mixtures. For most of the molecules, there is no plausible theory how they could have formed. For example, ammonia, there is no chemical hypothesis, which is like, even with experts in the field, accepted. And the person that asked the question is probably aware of the fact that as for oxygen, whether there was oxygen, lots of oxygen, not a lot of oxygen, that is debated a lot in the community. So indeed, it's also a mystery how those simple molecules and mixtures containing them came about. And I haven't asked the question, how did the particular composition come about? Like lots of oxygen or little oxygen, lots of ammonia, little ammonia, lots of other carbon sources, little carbon sources, carbon oxide, carbon dioxide, carbon monoxide. It's all a matter of speculation. But I like the speculating about cells better because, I mean, we have cells. We can investigate cells. We have them before us. We can put them under the microscope. We can analyze them chemically, biochemically. Whereas primordial Earth conditions under a hypothetical primordial Earth, we cannot really study that. This is something we can only infer from indirect evidence, mainly geological evidence. So I think personally, it's a better starting point to look at the biochemistry, to look at the more complex molecules and ask ourselves, how could they have come about? What can we know about their chemistry? Then I think we are on a bit safer ground than when we have to speculate about the conditions of a primordial Earth.
Well, that takes me to the very next question, which is how confident are origin of life researchers about the conditions of that early Earth? I suppose to add to that, if they are confident, what is the basis of that confidence? You mentioned geological. Is
Mhm.
that it? Is that it? What's the answer to that?
Yeah, it's basically geological. Of course, that assumes that when you investigate a certain how do you call that, level of geology, if you investigate a certain level of geology, that you have to sort of assume, okay, that is what it was like 2.5 billion years ago or so, whenever you assume that life first formed. So you have to have a time scale in geology in order to say, okay, this geological striatum, I think that's the word I was looking for, is the one that informs me. But researchers in this field are not unanimous, which is basically a good idea. It's a good idea not to be unanimous among researchers because nobody knows for sure. The theory of the origin of life will always be hypothetical because we cannot observe how it happened at that time, whenever that was, whether a long time ago or not a long time ago, whether God was in it or God was not in it. So it's good to not be unanimous because we don't know exactly, so we can test different hypotheses. But all the hypotheses, I think, don't work chemically, don't yield the result of, okay, this is the first step and we can connect that with a second step towards living biomolecules and third, it doesn't connect up. It's islands of chemical research that don't connect up.
Peter,
Mhm,
there's two questions that arise from that. I'll ask the first
mhm,
to hear your answer and then the second. The first is this.
mhm,
You mentioned in your talk that there are the people that continue to do chemistry, and there are the general public, if you like, whose knowledge of chemistry is somewhat limited or goes back to their formative days in school.
mhm,
Why do you think it is, though, that I would suggest the vast majority of lay people with no chemical
mhm.
understanding or background are under the impression and belief that scientists know how life started?
I have to be a bit direct answering this question, I'm afraid. This goes back to mass media, newspapers, um, overstating investigations and results from origin of life research. There is so much wrong things in the media and spread or insisted upon by journalists, which is not saying that I'm absolutely admire the profession of a journalist, but when it comes to origin of life journalism, there is such a desire, or I don't know, inclination perhaps to overstate results. Sometimes you read something like, it was proven that something in origin of life. And mind you, being a pharmaceutical medicinal chemist, this very much reminds me of overstatement of medical investigation. Very often people in mass media are like stating something has been discovered against this or that disease, which is just the first step, or perhaps first step of 20 more to go. So there is a kind of oversimplification. It's an oversimplification driven by the desire to make things simple. But the simplification also leads to the impression that people have almost discovered how first cells or life originated, which is simply not true. If you look at the expert journals and what the experts have to say, and I've quoted some in my talk, they know they don't know yet.
Well, that word "yet"
Mhm.
leads nicely on to my second question, which is this:
Okay.
the inference,
Mhm.
if not expressed, conclusion from what you're saying is that chemists currently know of no natural mechanism that explains the origin of life. Now, if that is an accurate statement insofar as you're concerned, then the word yet, the relevance of that is this. Are you saying that the state of science now is that we don't know of a natural mechanism, but we can keep looking for one and we may still find one? Or is the science and the chemistry advanced enough for us to state that no natural mechanism could ever explain the origin of life?
If you include among natural mechanisms that there is some kind of creativity which we haven't discovered yet, then I would say yes. But let me answer the question basically in a different way. I was asked this question when I debated a chemist publicly on the topic of origin of life, and he was of the opinion that we will discover a natural mechanism and will not need creativity or anything like it. And I was obviously of the other conviction. Now he said, look, the field of origin of life research is not very old, 50 years, 100 years, no matter where you want to set the beginning. So he said in another 50 years, we will know more and we will have found out how this could have happened by itself, meaning without any plan, design, or somebody to attend to it. So what I answered was that neither you nor me can know what science will have found out in 50 years' time. So let's have a look at the trend. Let's have a look at the trend during the 50 years, or during 100 years, or since Darwin's time, because Darwin had this very short passage in his Origins book on origin of life. If we look at the trend during the past decades, it's this. The trend is we find out more and more about how complicated, how carefully designed, whatever the designer was, cells are. We find that they're not just jelly with a few boxes swimming to and fro in them. So the trend is that the more science we do, the more obvious it becomes that even the simplest bacterial cell is actually a very fine-tuned, complicated organism or construction. So the trend is that in 50 years' time, if the trend goes on like it has been in the past 50 years, through biochemical research, through origin of life research, you name it, the trend is that it becomes more and more implausible that all this could have come about by itself, because it's all even more complicated than we ever imagined even 20 years ago. So that's the trend. And that is why I'm confident that when we talk about this, David, in 50 years' time, perhaps, we will, I think, even be more convinced that this needed creativity. Let me use this word again.
If the trend is that the more knowledge and more information we discover, the more we know that it can't be the natural mechanisms that are posited, that would almost be the opposite of the often cited God of the gaps, wouldn't it?
Absolutely. Yeah. Science does discover gaps, meaning we know that we cannot go from here to here in one step, and perhaps not even in 5 steps. But this is, like I try to show in this talk, that we don't discover gaps, we discover evidence for, as you probably at C4ID would put it, design or something. Creativity, you name it, organization. That's what we discover. And of course, I can understand that a person that perhaps doesn't believe in God and thinks, okay, God is around the corner of this thinking, which he actually is, I have to be honest. And if a person doesn't want to go in that direction, I can understand that everything I would consider as the discovery of something which is not a gap, but an evidence for organization, for plan, design, and all that. That a person who isn't into this thinking will shy away from it. And I can only ask you to reconsider, if you shy away from this, to reconsider. I'm not talking about God directly, and this is absolutely not my intention with this talk, to prove that there is God, not even the Christian God. We need other evidence for that, if it exists, but that's not our topic tonight. But please reconsider. It's not gaps which we fill with hypotheses, but we discover through scientific evidence, especially when we look at the origin of life, we discover that there is something like creativity, organization, integrative planning and all that, which we absolutely need to put these cells together. That's not a gap. That's a beautiful, wonderful discovery, which is actually being made use of. Not particularly the origin of life research, but biochemical and physiological research has informed a lot of human inventions, which didn't treat the discoveries as gaps that have to be filled as quickly as possible with mechanisms that are mindless. We use the mindful organization which we see for constructing useful things and sometimes terrible things actually in our technology.
Okay. Well, Peter, I'm going to hand back to David Galloway now, but just to say a big thank you from me for a hugely informative and interesting talk, and for the questions you've answered. I don't know what's more impressive, the fact that your breadth of knowledge and understanding of the subject, or the fact that you can do it in a language which isn't your first language. I'm not sure which one of those impresses me more, but there we are. Thank you ever so much, and I'll hand back.
Much, David. Thank you very much.
I'll hand back to David Galloway now. Thank you.
Good. Well, David, thank you. And I absolutely echo those closing comments that you made. Let me just add my thanks to Peter for a very thought-provoking and beautifully illustrated talk. Great input, searching questions also from the audience. That was good. So thanks everyone for your engagement. Let me just say in closing that we're planning another monthly webinar, but this next month will be on Monday, April the 25th. And on that occasion, I've agreed to make the presentation. And it's very interesting because I'm going to be picking up the very theme that Peter has been beginning to discuss as he closed out that discussion. It's based on a book that I published recently. The book is called Design Dissected, and the subtitle is the Design Real? And I had a little bit of fun with that from my background, just as Peter has brought his chemical background, and mine has been in clinical medicine, physiology and biochemistry. And so I've taken a little bit of a tour from 16th century Europe to modern neuroscience, from death in 1800s in Vienna to the streets of Glasgow and the life-saving influence of people like Joseph Lister, and in Paris, Louis Pasteur, from early attempts at anesthesia in Boston to studies of quantum consciousness. So we'll have a chance to look at some of the material that I've put in the book, not all of it by any means, but it will be fun just to try and explore some of these things. And we'll pull together an array of, I think, easily digestible evidence to show that, just as Peter has begun to demonstrate, that design in life is no gap, it's no accident, it's not an illusion, as some famous scientists try to persuade us, that the evidence does seem to support the idea that design is real. And perhaps I'll take the opportunity of just challenging people who choose to believe something else and list the intellectual cost that you have to pay to sustain that view. So if you're skeptical, or if you're interested in these questions of science and faith, even from that point of view, from an apologetics angle, from a purely scientific evidence angle, then I hope you'll find that interesting. That will be Design Dissected on Monday, 25th of April. And as we conclude today, let me just encourage you to visit the C4ID website. Make sure that you register your contact details with us so that you don't miss any of the events or updates. And similarly, why not subscribe to the YouTube channel? And there you'll find the recordings of many of the previous sessions that I mentioned earlier on. And there's some fascinating material to explore within that library of recordings. So with that, I think we sign out for today. Thanks again to Professor Peter Imming and to David Williams. That's it until the next time, and we look forward to seeing you on 25th of April. So until then, stay safe and thank you very much.